This is a presentation on glucagon gene expression in the vertebrate brain by Carlos Velez, Richard Wright and Claudette Ramlal. The source is a paper by Daniel J. Drucker and Sylvia Asser in the Journal of Biological Chemistry. The aim of this presentation is to review Drucker and Asser's paper that reports evidence for glucagon gene expression in the vertebrate brain. This slide depicts the different stages of glucagon gene expression. Starting with the glucagon gene on the left, we first have transcription to yield messenger RNA, after which follows RNA processing including polyadenylation, capping and splicing. The RNA is then translated to the peptide proglucagon that undergoes post-translational modification such as cleavage, to yield a number of peptides including glucagon-like peptides, glycentin and glucagon. Prior to this paper, there had been evidence for glucagon and glucagon-like peptides existing in the brain, but it could have been that they were synthesized elsewhere and transported to the neurons. To test whether the gene is expressed in the brain, the scientists looked for evidence of glucagon mRNA transcripts in the neurons. The findings of this paper, published in 1988, were significant as prior to this there had been discovery of the expression of other genes coding for peptide hormones in the nervous system, and it had been proposed that these had roles in neurotransmission, neuromodulation, and as releasing and inhibiting factors in the brain. It had also been proposed that glucagon and glucagon-like peptides had roles in the central nervous system, but without the supporting evidence of proglucagon synthesis in the brain.
The key findings of this paper are the glucagon gene is expressed in the brain stem and hypothalamus with a messenger RNA transcript identical to the one synthesized in the pancreas and intestine translating proglucagon-related peptides in the brain. Glucagon messenger RNA transcripts were detected using Northern blotting, and the next slide will describe the methodology. The process was first carried out with total cellular RNA and then with polyadenylate RNA taken from different regions of the rat brain and rat intestine as detailed in the next slide. RNA fragments were size fractionated by electrophoresis and agarose help, then blot onto a nylon membrane onto which they were fixed with a UV light. Pre-hybridization to blot no specific size was carried out overnight with salmon spare DNA, followed by hybridization with P-level complementary DNA probes for glucagon, somatostatin, and cholecystokinin. Detection of the transcripts was then carried out via autoradiography. The total cellular RNA samples yield no hybridization glucose mRNA transcript except for the brainstem. The process was then repeated with the polyadenylate RNA, the result of which they are shown on the next slide.
So, moving left to right, we're looking here at the autoradiography results from the blots hybridized firstly with glucagon cDNA probes, then somatostatin, then cholecystokinin. The exposure was for 7 days. A strong signal revealing mRNA transcripts for glucagon was detected for adult brainstem, a very faint signal for fetal brainstem, and no other convincing signals apart from that for intestine. It's worth noting that the glucagon transcript detected in adult brainstem was only slightly larger at approximately 1300 base pairs than intestinal glucagon mRNA, that we already know exists, detected at 1250 base pairs. Somatostatin and cholecystokinin are neuropeptides known to be abundantly synthesized in the vertebrate brain, and cDNA probes for these were used to check the integrity of the hypothalamic and brainstem RNAs. The northern blot was rehybridized with these probes, and the mRNAs were shown to be abundant. Although the signals for hypothalamus were not convincing, there was a faint suggestion of a signal, so it was decided to try and provide more evidence for the mRNA transcripts in rat hypothalamus. another northern blot was run for hypothalamus and brainstem with longer exposure, and the next slide shows the results. As you can see, after 20 hours there is a band showing glucagon mRNA transcripts in the brainstem sample, but nothing for hypothalamus. But exposure of the same blot over 11 days showed a band in the hypothalamus lane at the same location as that for the brainstem lane. Presence of somatostatin transcripts again served to verify the integrity of the RNA, and the virtually identical amounts for hypothalamus and brainstem are consistent with the results from the previous blot. This demonstrated that the glucagon gene is expressed in the hypothalamus, just at lower levels than in the brainstem, and it has been detailed that these levels were approximately 100-fold lower. Following this experiment, a human neonatal brainstem cDNA library was screened with a rat pancreatic preproglucagon cDNA probe. 4 clones were obtained, and the sequence matched entirely that predicted from the genomic sequence of the human preproglucagon gene. The sequence contained the entire coding sequence for glucagon, as well as a signal peptide, glucagon-like peptides 1 and 2, and glycendin. To test whether the glucagon mRNA transcripts were translated in brain neurons, brain tissue was examined for glucagon and glucagon-like peptide 1 immunoreactivity, and results are shown on the next slide. As shown, GLP-1 immunoreactivity was detected in neurons in the medulla oblongata and hypothalamus, suggesting gene translation does indeed occur in these neurons.
To summarize, through the discovery of mRNA transcripts, the isolation of cDNA clone encoding proglucagon, and detection of GLP-1 immunoreactive irrelevant neurons, Ducker and Asa successfully demonstrate that glucagon is expressed in the brainstem and hypothalamus. This was therefore an important paper to promote the continued research into the target sites of the action on physiological relevance of these neuropeptides.